So welcome everyone, and thank you for joining us for today's webinar. The webinar's title is Leaf the Graph Paper Behind, Real-Life Applications for Leaf Area Meters. So today we're going to be talking about two of our offerings at CID Bioscience. The first is the CI202, and then the other model is the CI203. So just to introduce myself, my name is Galen, and I am an application scientist here at CID Bioscience. I have a bachelor's degree in biochemistry and a master's degree in food science from Michigan State University. I'm an IFT certified food scientist, and a lot of my previous experience is in the analytical field. So I worked at an analytical food laboratory as a manager. I also did food safety consulting for the food manufacturing, produce, and cannabis sectors. I'm really excited to be able to bring some of my expertise to the CID Bioscience. So quickly, let's go over today's agenda, what we're planning on talking about. We started with the overview. I already gave the introductions, but we'll go over some quick CI 202 and 203 overview, some features, some specifications, and then we're going to jump right into the research, the current research. People that are using the CI 202 and 203, and how they're utilizing that instrument to further their research. And then unfortunately, we had planned for some guest speakers today. Unfortunately, we were having a lot of connectivity issues due to their location being in Africa. We were having issues with being able to connect them onto Zoom. And so unfortunately, we are not going to be having any guest speakers today. So after the research, I will do a quick summary, and then we'll jump right into the Q&A. So this will be a little bit of a shorter webinar. But that's just how it's going to have to be. Sorry, guys. So a little bit about our company. CID Bioscience was founded in 1989, and so we have more than 30 years of experience building plant research instrumentation that can serve the research community as well as commercial needs. And so what we do is we create non-destructive measurement tools that will help researchers acquire more consistent and high-quality data. Our instruments are known for their ability to produce instant accurate results. All of our instruments are field-ready, durable, portable, with full data transparency. And we're really proud of the fact that we do all of the engineering, testing, manufacturing, all under the same roof at our headquarters in Camas, Washington in the United States. So let's jump right into an overview of these two instruments. So as you can see on the photos on the right, the CI202 is a pallet style instrument, and the 203 is more of a wand style. So with the 202, users are able to collect both non-destructive and destructive leaf area measurements. So how it works is that you would put the leaf under a transparent sheet on the pallet, and then slowly move the scan head down the leaf where a laser will shoot down. It's a 627 nanometer laser, and it will keep taking measurements as long as the width is non-zero. So whenever the lasers determine that the leaf no longer has any width, that means that the instrument will stop its scan automatically. So there's no programming, no calibration like that needed. It is a durable and self-contained instrument. You can use it in the lab or in the field. You can use it with virtually any type of leaf, needle, or seed. And you can store more than 8,000 data points on the device and then export that data for further statistical analysis. The 203, being a wand style, is an even more portable option. You can rapidly perform non-destructive measurements with high resolution, and you can do it on plants, living plants, in any environment really. And so it's a single hand operation. It has unlimited data storage, essentially. So we utilize an SD card as the data storage device in the CI203. So you can have as many SD cards as you want on you if you plan on taking hundreds of thousands of measurements at a single time. But it also is a perfect field instrument, but also a good lab instrument, because we do have a optional conveyor attachment that will allow you to take a rapid set of measurements of detached leaf samples. So if you already harvested some leaves, you wanted to rapidly churn out, 100 measurements. We do have a conveyor attachment that makes it really simple to use and get that data very quickly. So going over some features of the 202 pallet style leaf area meter. So the parameters that it measures are area, length, width, perimeter, and then it also will calculate shape factor and ratio. So the measurements can be non-destructive if you keep obviously if you keep the leaf on the plant, but it also can be used in the laboratory or in a setting where you have already harvested those leaves in a destructive way. It is able to flatten those curled leaves so you can get the really precise measurements. So we have that transparent sheet, kind of flattens down the leaf for you so that you don't miss any sections of the leaf and get inaccurate measurements. Our resolution for the instrument is 0.01 centimeters squared. It's a really simple, straightforward operation with no calibration needed. It's really, and it has as I mentioned just a little bit ago, it automatically can detect when the leaf is no longer being measured. So there is no, telling it when to start, telling it when to stop. It'll automatically detect that for you. It is a really lightweight device and all self-contained. So it has the built-in data logger and then a rechargeable battery and USB ports for the data transfer to the computer and also for charging the device. The 203 is very similar in that it also collects the measurements of area, width, length, perimeter, shape factor, and ratio. But the 203 can also do void counts. So if you have large holes in your leaves, it also will count those voids. It can be non-destructive, or as shown in this image on the right, with the conveyor attachment, it can be used if you've already taken a destructive sampling. The conveyor is really good for those rapid destructive measurements. And the wand itself has a heavy kind of arm to it, so it'll flatten those curled leaves out so that you're getting an accurate measurement. It is a very lightweight instrument, very portable. The graphic display on the LCD display, it will show a leaf outline just to verify that your scan was accurate and actually took a scan of the entire leaf. We do have GPS tagging on it so that you will get location data for every measurement. As I mentioned, the SD card for storage, and for data transfer. So virtually, as long as you have multiple SD cards, you can essentially store as much data as you need. It does have USB charging and USB transfer of data as well as a backup with the rechargeable battery. And so the maximum thickness is 14 millimeters for leaves, and then also 150 millimeters wide. You can increase that width by using the conveyor. So if you have really, really wide leaves, then the conveyor might be the best option for you. But just like with the 202, there's no calibration necessary. So both of them great options. They essentially measure the same thing. We just wanted it in two different formats so that our users had options. So just some applications. Obviously, a lot of plant physiologists will use the CI202 or the 203 to relate changes in shape characteristic to physiological function. As we'll see in one of these studies, actually, I think the first study I'm going to go over, the instruments can be used for agricultural purposes. So looking at soil or plant treatment efficacy, or looking at orchard management or field management strategies. And then also can be used in botany for looking at phenotypic changes, and also advancements in technology, as we'll see later on in a couple of these studies that look at kind of modeling phenotypic changes and phenotypic plasticity in these plants. So some applications you might not have thought of, which we've seen users utilize the CI202 and 203 for, are looking at things like seeds, wings. We actually had a user using this instrument to measure the area of a bat's wings. So while the bat's still alive, putting the wing underneath the plastic sheet and scanning it to take an area measurement of the wing of the bat. You could also use it for things like insect wings if that was something that interested you. And then even inorganic materials like manufacturing components. So it's really a really versatile tool. It's a laser scanning area meter, essentially. So let's dive into some of these studies. So I've got 6 studies for you guys, and they'll cover a kind of a wide range of uses. And I'll just kind of go over briefly what the experiment was about, show you how they used our instruments, and then show you some of the results, and then we'll move on to the next study. So this first study is an agricultural based study. So essentially this study, which is titled Effective Orchard Floor Management Practices on Growth, Yield, Leaf and Soil Nutrients in Nectarine. This study was conducted in a northern state in India in the Himalayas. And so they were looking at basically trying to transition from peach to nectarines, because the peach crops have a less of an economic return. But they're also concerned about all the different, the varying conditions of the Himalayan region. And so they're kind of worried about how much water the plants were getting, or the trees, the nectarine orchards were getting, because it was really variable. And so essentially what they looked at is different ways to kind of consistently ensure moisture in the ground for these trees, even though the rain in the region can be very unpredictable. And so they essentially just looked at a bunch of different soil treatments, right? So different mulches, and then as well as some weed controls, and then just a standard control treatment. And so what they utilized the 202 for was actually looking at leaf area. And so what they found was that different orchard floor management practices significantly influenced the leaf area and the total leaf chlorophyll content of the nectarine. So they, looking at this table here, essentially what they did is with looking at another variable that's a good indicator of tree health. They were looking at the leaf area. And so with the different treatments, you can see obviously the treatment number 1 with the black polythene mulch yielded the highest leaf area, or the second highest leaf area, I should say. The treatment number 2 with the bicolor mulch, or bicolor polythene mulch, a very similar leaf area to the first treatment. And then significantly smaller leaf areas as you go on down their other treatments, and then the control being the lowest leaf area. So what they basically were concluding is that, with these, black or bicolor polythene mulches, you not only do a good job of retaining moisture in the soil, you also do a good job of kind of reflecting that certain red to near infrared range of sunlight back to the leaves, which will increase their chlorophyll content, and generally improve the health of and well-being of these nectarine trees. So basically what they want to do is try to find the best kind of management strategy in order to more successfully grow these nectarines in a region with unpredictable climate conditions. And they did a pretty good job of determining what type of flora management practice would be best to ensure the maximum yield and maximum health of these nectarine orchards. So really cool application for an agricultural field. The next study is actually a dissertation that I found that I thought was really, really interesting. And so this is a PhD dissertation for Ronghai Hu at the University of Strasbourg. And the title of this dissertation is the Consistent Forest Leaf Area Index Retrieval Using Ground and Airborne Data. So this is a 150-page dissertation, essentially is trying to determine the best way to calculate leaf area index. So only a small, of a small part of this dissertation was actually utilizing the CI 202 or 203, but the reason they were using it was really interesting to me, because essentially they were trying to determine the best indirect methods of measuring leaf area index. So there's hemispherical photography and other and lots of other transformations to put on, like gap fraction analysis and stuff like that, in order to get the most accurate leaf area index from either ground instrumentation or above ground, like LiDAR based instrumentation. And so at the beginning of this study, essentially it was a review of all the technologies that were currently available at the time of the study in order to determine leaf area index. And so the most direct way to relate it to, instead of having to measure everything manually as the direct measurement of the leaf area was actually the CI 202. So they utilized the 202 to basically measure this representative sampling of leaves that were collected, 2,974 leaves to be exact, which is a lot of leaves to measure, but didn't take that long with the CI 202. And then once he was able to determine the leaf area, he was able to determine the leaf area of one side of the tree using the equation there on the right side, basically taking the leaf area of the sample divided by the weight of the sample multiplied by the weight of all of the samples and all of the leaf samples. And so by getting the leaf area of the tree, he was then able to get the leaf area of the entire region by further extrapolating and using more mathematics. And from there, using that as kind of the established direct measurement, the defined leaf area, doing it the classical way, as classical as can be, but basically speeding up his process by utilizing the 202 instead of having to take a ruler out and measure every single parameter of these leaves. And so when he was comparing it to some of these indirect measurements, it was finding all sorts of variation in the indirect measurements of the leaf area index as compared to the CI 202 direct measurements, and later concluded that there's inherent flaws in all of these different indirect measurement technologies, because they aren't taking into consideration mathematically things like clustering effect and things like the light influence, at different times of day, and sort of all of these different variables that aren't being taken into account eventually lead to a difference in so the I don't have the number in here, but the leaf area as verified actually by the 202. So he did two different studies. I mean, he looked at this initial sampling of 2,974 leaves, but then he went and he did it again with a little bit of a smaller sample just to ensure and verify that this leaf area that he got was indeed a consistent number. And both times he got 3.81. So looking at this table down here, you can see that a lot of these vary quite differently from that true value of leaf area index. So going forward in this study, essentially, he developed the most accurate and thorough, I should say, method for calculating leaf area index indirectly using both data from a instrument on the ground as well as LiDAR data from above ground, and to get the most accurate possible leaf area index completely indirectly, but basically utilizing the 202 as his kind of direct measurement method that would, that he would be able to reference back and look at in comparison to these indirect measurements. So really cool dissertation. I would recommend checking it out if you have a lot of time on your hands. It is quite long, but also very interesting review of and application of all the different technologies out there for leaf area index. So the next study I have is investigating basically irradiated and non-irradiated sewage sludge as a treatment for soil. So looking at changes in growth characteristics, chlorophyll, macronutrients in basil plants as they are treated with sewage sludge that is both irradiated and not. So why irradiate the sewage sludge? Because that's becoming a really popular option for this type of waste in order to essentially, kind of sterilize it, make it more safe. And so they wanted to see if that irradiated sewage sludge has an influence on the growth of basil plants, as kind of an indicator for other plants out there. And so they looked at multiple different concentrations of gamma ray irradiation, as well as different amounts of sewage sludge per kilogram of soil. So what they did is they looked at, they used the 202 to look at leaf area as one of their indicators for plant growth and plant health, as most people usually do, and found a really interesting and strong response to the actual irradiated sewage sludge soil. And as you can see in the graph on the bottom right, the column that is marked 20 kgy, that's 20 kilograms of gamma ray irradiated soil. So those columns are obviously much for the 15 and 30 grams of sewage sludge per kilogram soils. Those highly irradiated, sewage sludge samples had actually the best growth characteristics for, especially when it came to leaf area. So it was an interesting thing to see that, but then obviously once you get too much, you can see from the graph, there is a threshold where too much sewage sludge is a bad thing. So it needs to be mediated, and you need to make sure that you are at a specific concentration of sewage sludge. But again, really cool application for this is a problem for a lot of people around the world to encounter lots of sewage sludge in their environments, and so being able to treat it with by irradiating it so it is not harmful to as harmful to humans or wildlife or plants, and then being able to actually utilize it to assist growth and health and yield for crops and for other plants. So I thought that was a cool application, just because how often do you get to see studies about irradiated sewage sludge? So our fourth study that I'm going to go over actually starts to look more into the how the 202 and the 203 can be utilized to actually look at even further technological advances. So this study was essentially a look at how can we model the a plant, the eucalyptus tree, how can we model it by using artificial neural networks so that it can predict things like phenotypic plasticity, even when it's tightly clumped together or when it's super spaced apart. So they wanted it to be a really versatile model that they build that can predict the biomass of the eucalyptus based on things like leaf area, specific leaf area, and then also by other parameters that they measured. But they really focus on specific leaf area as their kind of variable or indicator parameter to actually do the prediction for the biomass prediction. So they wanted this robust model. So this is something that we can relate to at CID Bioscience and Felix Instruments, because this is something that we do as well. But doing this modeling process, so trying to trying to make a model that can consistently predict wood biomass in eucalyptus, regardless of all of these variables that are thrown at you in nature. So they wanted to do it first to kind of account for all these different spacings between trees. And so what they found is actually that they could create a really robust model just by taking these measurements, these specific leaf area measurements, which they got from doing leaf area and leaf perimeter measurements with the 203. And so what they did was essentially at 48 months, 60 months, 72, 84, 96, and then right before, or between, I guess it was between 96 and 108, probably at month 100, they took all these measurements of these leaves, and they basically collected 10 leaves in each third of the canopy. So each upper, middle, and lower part of the canopy, they would collect 10 leaves, so 30 leaves per canopy, and then they dried them to get dried them at 65 degrees to get biomass. But also took all the leaf area parameters with the 203, and then calculated the specific leaf area based on both the leaf area and the dry mass of the leaves. So basically, what this is showing with these different colored dots on the left is all the different spacings that they investigated. So all of these dots represent a different category of spacing between trees. And so, as you can see, the trees that are spaced closer together ended up actually having higher leaf areas or leaf perimeters and leaf areas, mostly due to competition, trying to fight for who's going to get the most light. And that number decreased. So once they took enough data points, they could teach their neural network, this is, with these different spacings, this is the trend you're going to see with leaf area, leaf perimeter, and specific leaf area. And then this is what it's going to, and then that trend is going to translate then to the biomass of the eucalyptus trees. And so they did end up building a pretty successful model to be able to predict these things. So that's utilizing the current technology to create advancements in technology, and that's something I always get excited about. So another really cool study coming out of Brazil. And so I think we can be looking forward to more artificial neural networks, deep learning based modeling kind of in the near future here. So the fifth study that I found that I wanted to look at, just because it really caught my eye, is this study that looks at 3D visual modeling of super rice, and using that 3D visual modeling in order to help predict changes in the leaves, phenotype, based on the agricultural practice and the agricultural manipulation. So they're looking at two different practices: the farmer's practice, which is FP, and the super high yielding practice, and looking at how these how the phenotypic plasticity is changing between these two practices of the same type of rice, the same cultivar of super rice. And so just because of these two practices, they were seeing these large differences in leaf area index, and then they were using that to help build the data collected of leaf area index, and the changes that they were seeing were used to help build this model that could then be used to help predict these changes in further analysis. So they essentially basically digitize these plants, as you can see in the left image, these super rice plants, and they destructively sampled them after they did their field measurements. So they did all their aerial length, maximum width of the blades, and they did that all with 203. And then you can see in this graph down here is where they're actually plotting their differences in cumulative leaf area and leaf area index between the two practices. So the open circles are the super high yielding practices, and then the solid circles are the farmer's practice. As you can see, as the height increases of the plant, there are pretty significant and drastic changes in their cumulative leaf areas and leaf area index between the two practices. So this is something that I think is a really cool application of using our instruments. Again, trying to further technology by using our instruments to collect data that can then be used to create this new technology that can do the modeling, and in order to then in the future predict changes in phenotype, when new agricultural practices or agronomic manipulation comes into play. So another really cool study, and this one's a really for those that are technologically inclined, I definitely recommend you check this one out, as well as the neural network study, because this was a really cool one to read. And then to finish it out here, we have a really classical use. I wanted to show kind of the classical use for this instrument of just looking at how a variable like light availability can influence the leaf and crown morphological characteristics in a species of oak tree, essentially. So it's really, this is kind of the really basic use case that we, the first one that came to our mind when you think about leaf area meters, is just thinking about going out to a tree and taking some leaf area measurements to investigate why something is causing the leaves to change in shape and size. And so it was just looking at a really good study, a solid study, very good design, and very good data transparency and collection process. So they utilized the CI202 portable leaf area meter, and they looked at the leaf size, so the area, perimeter, length, width, and then the length to width ratio. We call it the aspect ratio when it's the length to the maximum width. And so then they looked at how that changes with varying amounts of light availability. So depending on where these populations are. So there was 5 unique populations. So each one's going to have, varying levels of light, depending on how dense they're, or how close together the spacing is of the trees, how dense the canopies are, et cetera. And so, some takeaways are, and most of you who are familiar with plant physiology probably already know, but as leaves were competing for sunlight, they actually tend to grow so that they can try to capture as much sunlight as possible. So when there's not a lot of light available, they tend to be a larger leaf area so that they can kind of beat out the competition. So, but still a really good, robust study, and a really good example of just an easy way, a way to make your life easier if you're doing these kinds of studies where you need to take lots and lots of leaf samples, lots of measurements, and you want to be able to do it, without thinking too much. Then honestly, the CI202 pallet style and the 203 with the conveyor, if you've already harvested those leaves, it's a no-brainer really, compared to taking a ruler out and having to write down everything. The time savings alone is worth it. So, and that kind of rounds out all these studies. So everything from agriculture to modeling and artificial neural networks and classical, forestry applications. So this, and then also looking at the sewage sludge study, which was really cool, new applications, new types of soil treatments and stuff like that. So lots of different uses out there for the CI202 and the CI203. So in summary, there's, as I just said, lots of uses, lots of applications. And honestly, it's not just limited to the leaves. Like I mentioned earlier, if you have insect wings or seeds or manufacturing components that you need to get consistent, accurate, and quick results for area measurements, length measurements, width measurements, perimeter measurements, then the 202 and the 203 can be a benefit to you. So lots of researchers out in the field, either in academic or private or government sectors, can benefit from this because it's a really fast alternative. It's reliable with the laser scanning technology, and it's affordable. So really any researchers of plant physiology, of botany, of forestry, agriculture, all can benefit from using this instrument. And so both instruments are able to provide rapid destructive or non-destructive high-resolution leaf measurements. So you can use them in the field, you can use them in the lab. They're really versatile. They can store a lot of data. The batteries have long battery life. They're rechargeable. Data transfer is really easy. So it's just a really simple tool to use. And also, just a side note is that if you have any application-related questions, if you aren't sure that this instrument will work for your application, always reach out to either myself or our other application scientist, Eric Munoz Garcia. We are more than happy to assist you and help figure out whether or not our instruments will be able to assist you with your research. So, just really quick before we completely conclude, I want to talk about some of our offerings that we're releasing here soon. We're really excited about the upcoming release of the CI710 leaf spectrometer. So it's the CI10 SpectraView, and it is a drastically improved version of our current CI710 leaf spectrometer offering. And we are really excited about it. It will be coming out in the near future. We have all the built-in indices still for things like chlorophyll, anthocyanins, carotenoids, all those different reflectance indices and absorbance indices. So it does all three, reflectance, absorbance, and transmittance, all at once, all that data collected for you at the same time. We do have now added the capability of modeling, just like with the F7 Felix Instruments F750. You can do partial least squares regression modeling to create models for things like dry matter in spinach or things of that nature. So that is a new feature. You will be able to create those models, and we also are making it compatible with our Fruit Maps application. So lots of really cool, unique things, and we're really excited when we will have a webinar on it when we do the release. But just want to let you guys know that's coming up, and I want it to be in your wheelhouse. The other thing I really want to plug is on the Felix instrument side. We did just release our F751 Kiwi quality meter. I'm really excited about that. It was a project that I've been working on, and it is a portable, handheld, near-infrared spectroscopy device. It will instantly and non-destructively assess your kiwi fruit quality and maturity. So it measures dry matter and bricks in both G3 or Sun Gold and also Hayward varieties of kiwi fruit. So it's a really cool tool for quick and easy quality measurements and determining things like maturity and making harvest decisions out in the field. So, all right. If you have any questions, or we'll start with, if you want to get connected, here's all of our information for getting in touch with us. You can always email, if you have sales questions, email sales@cidinc.com. Go to our website. We will be, I think, updating that here soon as well, but we do constantly post updates on the website. And so that's the best place to go to see any sort of new product launches, updates to software or firmware or what have you, new applications, things like that. You can find art research articles that utilize our instruments by going to our website and going to the applications tab, and then searching for our instrument. Searching for a specific instrument, you can find a list of publications that have used our instrument. And then also, of course, these are all of our social media outlets. Follow us to see updates of where we are, conferences that we're at, or places we are visiting. If you or if you're in the area and you want to stop by, always reach out to us, and we are more than happy to host anyone that happens to be in our neighborhood, and give you a tour of our facility. And then, so that kind of concludes our webinar today. So if you want to quote, we will post this link in the chat, because you will not be able to click it through this webinar, but in the chat box you will. And so if you want to get a quote for one of the leaf area meters, just click that link and it'll take you right to the quote page. And then now I'm going to go ahead and answer you guys' questions. So give me a second here while I open the Q&A box. All right. So let me make sure. I don't want this to be blocking too much of anything. All right, so first question is, how can I use the CI203 to measure insect feeding? So if you're wondering, like, how can you use it to measure the actual voids in the leaf can be measured with the 203. But if you want to see changes in leaf area over time, which is what I'm assuming you mean by insect feeding, as opposed to actually measuring the insects themselves, if you're looking at how the insects are feeding on a specific type of leaf, and you want to leave it attached to the tree, then you just need to make sure that you're using the 202 or the 203 in a non-destructive way. So if you're using the 202, you would just simply hold it up to wherever the leaf is positionally on the tree, put that transparent cover over it, swipe the scan head, and it'll measure the leaf area. And you'll just want to measure that over time to see the rate of feeding, I suppose. It would be a good way to do that. But if you have a more detailed description of that, if I would appreciate it if you would email me, and then I will look into it further for you. But that's just kind of my off the top of my head idea for how you could use it to look at insect feeding. All right. So the next question is, in which cases does someone prefer the CI202 versus the CI203? So really, I mean, like you've hopefully gotten the idea from this presentation, is that they're both very similar. They utilize the exact same technology, essentially, to accomplish the same goal, the same measurements. And so what most people do is they say, do I need to do this non-destructively, or do I need to do this, or can I do it destructively? And if you're doing it non-destructively, most of the time I would say people prefer to use the CI203 because it is a little bit more of a convenient design for going out in the field and doing portable, on plant analyses. If you know for a fact you're going to be harvesting all the leaves already, and then it might just be easier to have the 202, and all you have to do is use the palette style, take all your measurements, that way. And so really it's just, I think that's the number one question that you have to ask yourself is destructive okay? Or is this something where it's imperative to the study that I leave the leaf on the plant? And then give me 1 second before I measure the next, or before I measure, before I look at the next. I'm going to go back to the beginning of the slideshow really quick, just so you guys can get a idea of the instruments here. We'll go to this one. And all right, now I'm going to get back to the questions. So the next question, can it measure greenness of leaf and disease affected area of leaf? So no, the 202 and 203 do not do disease counting, and they don't do chlorophyll content of leaves either. They just are specifically for area, perimeter, width, length, and then from there aspect ratio, and as well as shape factor. And the 203 does have the added bonus of being able to do void counts. So that is something, if void counts are important to you, if a lot of your leaves are not fully intact and have voids in them, then the 203 might be a better option because it'll give you a more accurate measurement of your leaf area. The next question is, can it count the number of seeds and its length, width, and volume? So if you have multiple seeds, then it would not be able to tell you how many seeds there are. You would want to just do one seed at a time, or do multiple seeds and then all together. And then since okay, so let me backtrack a little bit. If you know the number of seeds you want to put on the palette, then I would recommend actually putting that number of seeds on the palette, scanning it, getting that total area and length and width and volume, then dividing it by the number of seeds. But it's not going to be able to. It won't be able to actually count seeds for you. If you have a random handful of seeds and you put it under the transparent film and then try to scan, it's not going to tell you how many seeds are underneath that transparent thing. But it will give you seed length, width, and volume if you do a single seed at a time, or like I said, if you have a bunch together but you know the total number of seeds, then you could use that to calculate your individual seed length, width, and volume. So the next question is, can it observe and make difference in shape of leaves and seed? So yes, so the 202 and 203 do look at, they are able to calculate shape factor. The 203 also is able to give you a outline image of the leaf or the seed that you are scanning. So what I would say is that it absolutely can observe and tell between shapes. You just want to make sure that you aren't scanning something that's too big for the scan screen. That's all I. That's probably the only warning I have, is that we do have a 15 centimeter maximum width. So for both of these instruments, that width does get wider, as I mentioned, for the 203 if you get the conveyor attachment, but still in general, you will always be able to observe a difference in shape of the leaves when using these instruments. So yeah. So the next question is, any seed applications? So as I mentioned, I personally I haven't seen any studies. I know there probably are some out there that utilize our instrument for seeds, measuring seed size and area, length, width, perimeter, all that. But I personally don't know. I will do some research and get back to you. If you could send me an email, please, and let me know, what you're interested in as far as seed applications are concerned. I can look into that for you and get back to you on that. The next question is, what's the smallest leaf that it can measure? So we have a resolution of 0.01 centimeters squared. So I would say don't try to measure a leaf that is smaller than 0.01 centimeters squared. And then anything bigger than that should be able to be measured. I will say that it does look for, for the CI202, the scanner is looking for a change of 1 millimeter or less. So if it's scanning and it sees that the width has gone from 1 millimeter down to something less than 1 millimeter, then that's when the scan head is going to decide to stop scanning. So when it says it's non-zero, when it says it's zero width, that's when the scan head stops scanning. So if it's too small for the resolution, I should say not 1 millimeter. Sorry, that's 1 millimeter is every time it takes a new measurement for width. So if you have a size or a width that's less than 0.01 centimeter or 0.01 centimeters, then yeah, I guess I would 0.1 millimeters. If it's less than that, I would not recommend measuring that because it's not going to work for the CI202. But the CI203 has a different mechanism for determining when to stop scanning. Although the resolution is still the same, 0.01 centimeters squared. So don't try to measure a leaf smaller than that in the 203 either. But the CI203 uses an optical sensor to determine when motion has begun and stopped. And that's how it knows when to start and stop scanning your leaf. So the next question is, how can I measure how much an insect fed? So yeah, so I already kind of went over what I think this question means, but if you're measuring change of any sort of change in the leaf shape, structure, width, area, anything like that over time, these instruments are totally suitable for that. So you can just take multiple measurements throughout the, a series of time when insects have been, feeding on the leaves, and then you can see the decrease in area, the changes in the perimeter, changes in length, width, things like that. So, so I would say it doesn't give you any sort of direct insect feeding measurement. If there is an index for that, I'm not aware, but it does give you an indirect way to measure that. So is it safe to use with leaves that leave traces on gloves and instruments like tobacco, tomato, cucumber, zucchini? So asking, is it clean, easy and safe to clean? Yes. So that's also a reason, another reason for the transparent sheet. You can always get more of those transparent sheets from us, and we will always send you more. As far as cleaning it goes, yep, very easy to clean. But you know, you may notice over time that you scratch it up enough that you feel like you need to get a new one. So then we always have more we can send you. You can always get a new one to refresh it. They're really easy to change out. So yeah, I would say totally safe to use on any of those commodities. The next question is, how long is battery backup? So actually, okay, so really quick, going back to that question I just answered, we do provide you with 2 sheets initially. So when you get it, you will have a spare, you will have a backup sheet in your with your instrument, as well as you'll have a cloth to be able to, like one of those like microfiber cloths to be able to clean off the sheet as well. So that comes with the purchase of the instrument. The next question is, how long is the battery backup? The battery lasts, I want to say something like 5,000 measurements or something like that. I'm not 100% certain. You know, we have all the specifications up on our website. If you go to www.cid-inc.com, and then you go to the products page and you click on 202, CI202 or CI203, and then there will be a specifications tab, and each one of those will be able to provide you with like the things like battery life and all that, and all those kind of specifications for the instrument. I'm not 100% certain off the top of my head how long the battery back life is, but I do know that it lasts for quite a while, for quite a lot of measurements. And then, what is the reproducibility and accuracy of data? That's a great question. I am fairly certain that when the leaf is, I think we did a study where if the leaf is greater than 10 centimeters squared, or the leaf length is greater than 10 centimeters, one of the two, our accuracy is plus or minus 1%. The next question is, what is the life of this instrument? I would say the life is mostly determined by your care of the instrument and how much abuse, I guess you could say, you put the instrument through. So as long as you aren't consistently dropping it and leaving it out in the rain and things like that, you send it in for routine, for maintenance when you need to, then you can make this instrument last as long as you need it to work. So I would say the life of the instrument is almost completely dependent on the user. And we have some that are in the field. I know that are somewhere around 8 to 10 years old. So it really does depend on, how well you take care of the instrument. So you can make it last that long, or if you choose to be a little bit more neglectful, then it probably won't last quite as long as that. So yeah, well, I think that is the last question, and I will go ahead, and if you have more questions, please feel free to email me, and I can put my email in the chat box as well. And then, we just got one more question really quick. Any discount for academia? So all these, for the sales questions and for questions of this nature about quotes and discounts, please reach out to our sales department, sales@cid-inc.com. Please send them an email, and they'll be able to answer that for you. All right, but yeah, I will throw my email into the chat box. If you have further questions, feel free about applications and things of that nature, please feel free to email me. For your sales related questions, like I said, go ahead and email the sales team. And then, yeah, thank you guys so much for joining, and I really appreciate your time. I hope everyone stays healthy and out there, and avoids this virus that's going around. So if you have more questions, reach out. But thank you guys for joining us, and we will see you guys for the next webinar.